Today, we shall discuss about peptides and proteins. So let's have a look at what peptides and proteins are. Proteins are linear polymers formed by linking the alpha-amino group of one amino acid to the alpha-carboxyl group of another amino acid. This type of linkage is called a peptide bond or an amide bond. The formation of a dipeptide from two amino acids is accompanied by the loss of a water molecule. The equilibrium of this reaction lies on the side of hydrolysis, rather than synthesis under most conditions. Hence, the biosynthesis of peptide bonds requires an input of free energy. Nonetheless, peptide bonds are quite stable kinetically because the rate of hydrolysis is extremely slow. So interestingly, the lifetime of a peptide bond in aqueous solution in the absence of a catalyst approaches 1,000 years. A series of amino acids joined by peptide bonds form a polypeptide chain, and each amino acid unit in a polypeptide is called a residue. A polypeptide chain has directionality because its ends are different. Alpha-amino group is present at one end, and an alpha-carboxyl group at the other. The amino end is taken to be the beginning of a polypeptide chain. By convention, the sequence of amino acids in a polypeptide chain is written starting with the amino terminal residue. Thus, in the polypeptide, glycine, serine, valine, alanine, cysteine, glycine is the amino terminal, N-terminal residue, and cysteine is the carboxyl terminal, C-terminal residue. An overview is given so you can relate the diagrams together. Now let me tell you a little something about how you can write the abbreviated form of amino acids. Each amino acid has a specific abbreviation. You can write it in a 3-letter form or as one letter of each amino acid. It is shown in the table below. A polypeptide chain consists of a regularly repeating part, called the main chain or backbone, and a variable part, comprising the distinctive side chains. The polypeptide backbone is rich in hydrogen bonding potential. Each residue contains a carbonyl group, which is a good hydrogen bond acceptor, and an NH group, which is a good hydrogen bond donor. These groups interact with each other and with functional groups from side chains to stabilize structures. Most natural polypeptide chains contain between 50 and 2,000 amino acid residues and are commonly referred to as proteins. The largest single polypeptide known is the muscle protein titin, which consists of more than 20,000 amino acids. Polypeptide chains made of small numbers of amino acids are called oligopeptides or simply peptides. The mean molecular weight of an amino acid residue is about 110 grams per mol, and so the molecular weights of most proteins are between 5,500 and 220,000 gram per mol. We can also refer to the mass of a protein, which is expressed in units of Daltons. 1 Dalton is equal to 1 atomic mass unit. A protein with a molecular weight of 50,000 grams per mol has a mass of 50,000 Daltons, or 50 kilodaltons. In some proteins, the linear polypeptide chain is cross-linked. The most common cross-links are disulfide bonds, formed by the oxidation of a pair of cysteine residues. The resulting unit of two linked cysteines is called cysteine. Extracellular proteins often have several disulfide bonds, whereas intracellular proteins usually lack them. Rarely, non-disulfide cross-links derived from other side chains are present in proteins. For example, collagen fibers in connective tissue are strengthened in this way, as are fibrin blood clots. Now, let's talk about trans and cis bonds in peptides. Do you know what is the difference between cis and trans? In the trans configuration, the two alpha carbon atoms are on opposite sides of the peptide bond. Whereas in the cis configuration, the two alpha carbon groups are on the same side of the peptide bond. Almost all peptide bonds in proteins are trans. This preference for trans over cis can be explained by the fact that steric clashes between groups, attached to the alpha carbon atoms, hinder the formation of the cis configuration, but do not arise in the trans configuration. Do you know what steric clashes means? They arise due to the unnatural overlap of any two non-bonding atoms in a protein structure is called a steric clash. This is all for today. For more videos, keep watching sqadia.com.